over to you.
Thanks so much, Vitaly and Sergey, and very nice to join you all, albeit online. So I'm going to just go through a couple of things in terms of where we've been looking at using polymers in applications across from vaccines and medicines. And I'm sure you're all aware there are some generic problems in treating difficult diseases. And so we've been looking at a range of different formulation parameters to address some of the key problems in difficult diseases. And what I hope to show today is that polymer structure is actually very important in therapeutic delivery. And actually, it was nice that one of the earlier talks actually cited one of our papers, which I'm going to talk a little bit more about later on polymer size and structure and architecture. So I'm going to give a couple of examples of polymer architectures and how the polymer science contributes to the efficacy of these particular materials and formulations in preclinical models of infectious diseases and cancers. Now, there are some generic problems I'm sure many of you are aware of, but many of our current therapeutics are suboptimal for cancers and infectious disease, and typically that's to do with therapeutic window. We don't always maintain the drug concentration within this key threshold, which is above the minimum effective dose and below the maximum tolerated dose. So many cancer drugs in particular have this very narrow window, and you have oral delivery, you have the danger of going above that. Sometimes an injection, it can fall away too quickly. There are also very unfavorable chemical properties. So here again, some anti-cancer drugs, but also things like RNA and proteins are difficult in terms of druggability. Many of our systems are not targeted to the disease sites in the body where they need to go. And an important point is that one can develop resistance to drugs if they're not dosed properly. One of the key issues that we face is that many anti-infectives and cancer drugs need to be injected systemically. That's partly a consequence of the narrow therapeutic window, but also for some of the other properties of drugs. And in order to do that, anything that goes in the body, of course, has to be filter sterilized, and the formulation must, of course, be stable in the bloodstream when it's in transit in the body. But also, it's got to be stable in storage. Sometimes these medicines are lying around for quite some time. So there are issues in terms of making sure these formulations can be used fully for the times that they're needed. And what I hope to show you today is that both transit in the body and across some biological barriers can be very dependent on the structures of the polymers or carriers that we use. So first of all, I'm going to talk about nanoparticle vaccine formulations. And obviously, I'm not going to go into any detail about this. I think you all know the issues there and how very successful mRNA vaccines have been. So the numbers are over 2 billion people. It's quite an extraordinary example of science and how science really has come to the rescue. But there are storage stability and cold chain issues for a lot of these current vaccines. And although Moderna have announced longer shelf lives for their other candidate vaccines, there are still some questions about whether you could store these kind of vaccines for a long time at room temperature, for instance. So how might polymer science enhance the solution to these problems? Now, one of the things about dealing with drugs like RNA or in this case, vaccine components, is that you can't simply inject free RNA. And that's because nucleic acids are degraded by enzymes in our body. So if you just do an injection, then our natural defenses will, of course, degrade the foreign RNA. That's part of the viral defense pathway. And mRNA on its own triggers immune sensors. That is, again, part of the body's response to invasion. If it sees nucleic acid sequences, foreign nucleic acid sequences, then that switches off a number of pathways and switches on other ones. Now, in vaccines, that can be advantageous if you get an immune response, of course. But if you hit the wrong receptors or the wrong receptor activated, that can actually cause some cells to shut down protein production altogether. So your vaccine wouldn't work. RNA would not be translated to the, if you like, the spike protein in COVID. And even if transported to the correct cells, and that's a big if in some cases, mRNA can't cross cell membranes efficiently. So you need a carrier to take mRNA into the right cells to produce an immune response or a therapeutic response. Now, the lipid RNA formulations have been very effective. Let's be honest, they've been quite extraordinarily effective. but they're not perfect. And for beyond vaccine applications, there may be some significant issues with some lipid formulations, which we're looking to overcome. Now, just a little bit on the lipids, just again, a bit of context. This is a computer simulation of what these lipid nanoparticles look like. The actual formulations, and particularly BioNTech and Moderna, again, very, very successful formulations, but there's a lot of stuff protected by patents and by know-how. And whilst I say there's computational predictions of these structures, there's the PEG layer, here's the RNA around this sort of sea of lipids and buffers. And this is what these things look like under an EM. There are a range of common components, cholesterol and so on, phosphatidylcholine, and usually a pegylated surfactant to stabilize the particle. So polymers are really important in our existing vaccines, and people talk about them as lipid nanoparticles, but actually the polymer, the pegylated component, is incredibly important. But there's also some proprietary ionizable and amphiphilic lipids. So these are quite interesting structures, and the details of why they operate are still slightly controversial. But so there are these pegylated amphiphiles as well. So these proprietary ionizable and amphiphilic lipids. There's a lot of things like cryoprotectants and buffers, and again, they have a role in formulation stability, but I don't have time to talk about those today. But as it turns out, the structures of these RNA lipid nanoparticles are very dependent on the preparation method. And again, that sometimes is covered by patents or by know-how, and it's not trivial to make these things. The other issue, as I mentioned earlier, is long-term storage at ambient temperature is not ideal for these systems, and shear and so on. So thinking about spraying these lipid nanoparticles may not be ideal either. So there are still some questions about if we want to have a non-injectable and inhalable formulation, then are the lipids the right ones to use? So formulating RNA inside a carrier is, sorry, I've just gone one too far. It's a simple example of nanotechnology. All you do essentially is you have RNA, multiple negative charges, and we mix them with some polymers, which are positively charged, or lipids, which are positively charged. So we have usually a soup of different lipids, as I mentioned in the previous slide. And what happens with the conventional lipid nanoparticles is the RNA goes in one line, lipid and ethanol solution goes another. They're mixed in particular ways. And as you change the solvent polarity more from ethanol through to water, we get this gradual assembly and collapse into these particles, which are coated by polymers, typically PEG lipid, which coat the precipitated surfaces, and that gives them some of their stability. And that's been known for some time, but the detailed structures are still not fully known. And so this is this example of how we do this in our lab. These are actually microparticles, but we do this with nanoparticles as well. So the process itself, as I say, looks very simple. Here's the positively charged systems mixing with the negative charged systems. And over time, eventually these build into a kinetically trapped particle, which actually looks a bit like a virus in terms of size and structure, but actually it's acting as a vaccine or a therapeutic. But what variables contribute to the success of these vaccines or therapeutics? And this is what we've been looking at, particularly over the last 3 years. And so this is work of Pratik Ganani, who did his degree and PhD at the University of Warwick. And I know Dave Haddleton obviously worked a lot with him. So Pratik is a fantastic polymer chemist and fantastic pharmaceutical scientist. He's really developed his skills, and he worked very hard on this polymer, Paybol, which is a microaddition reaction between this amine and this acrylamide. And this polymer has been shown by a number of people to be quite effective in delivering RNA. But actually the polymerization is not quite as simple as many people think. And so a straight microaddition would form this linear polymer. But the reality is, of course, that if you don't get this right, then you can get some level of uncontrolled radical polymerization. And that's exactly what Pratik saw. And did a whole set of studies about how these polymers are made. And as you can see under argon, we do end up getting this runaway molecular weight increase. And that's a result of this unintended radical reactions. And chain branching and so on. Whereas in air, if we inhibit that reaction, then we see much less in terms of a free radical reaction. We just get a typical polycondensation reaction. So Pratik then looked at a range of different polymerization temperatures. Nothing too complicated about this, but we can see, as you'd expect, as you go up in temperature, the molar masses go up for a particular time. And likewise, if you microwave, you can get to high molar masses much more quickly. Now, why does this matter? Well, as it turns out, if you then start looking at the expression of the transgene, so this is the protein, in this case it's luciferase, so it's an RNA encoding for luciferase. So we get more light production with some polymers than others. And as you can see, there are at least 2 orders of magnitude, in some cases 4 or 5 orders of magnitude difference between polymers essentially nominally of the same type and nominally of the same structure, but in fact with different molar mass distributions. And in fact, in these polymers, we found that the materials with the tightest molecular weight distributions actually gave the greatest transfection efficiency. So again, Dave Hamilton mentioned earlier that controlled polymerizations have not been perhaps as successful in terms of radical polymerizations in commercial applications. But if this were to be the case for these systems, then a RNA delivery polymer of tight molar mass might be the thing you'd go with. But the story is a bit more complicated than that. And we try to start disentangling some of the other questions around RNA delivery with some polymers that essentially do charge reversal. And again, this is something that Dave Hamilton, I know, and a couple other people at Warwick have been working on similar polymers, but for other applications. And we've been working on similar things for a number of years. And we noticed that some of these polymers were particularly effective at delivering DNA, not RNA. So Pratik basically made a series of polymers and varied some of the side chains, and then he put in this charge reversal group here, so that we go from a cation which binds to RNA to an anion that doesn't. So this was a way of releasing RNA, and we intended this to be a rapid release mechanism. Now, what you see is that, in fact, the different polymers, again, show different transfection efficiencies, and you can see a range of different overall luminescence here, but also a range of cell toxicities. And often you see that the most potent polymers are also the most toxic. When we did this work with Anna Blakeney and Robin Shattuck at Imperial College, we looked also at these polymers delivering RNA into human skin explants. And what was intriguing here was that we had really quite marked differences again between different polymers in terms of the number of human cells that they transfected. And we did see some differences in terms of the cell types. So whether it's T cells, of course important in the immune response, or just the epithelial cells which were targeted. So there were a range of different cell types targeted by these polymers, and it varied across the set. But this number here is kind of interesting because the percentage of cells that were transfected was about 10–12%. Now that doesn't seem very much, but that's actually quite a lot higher than one often gets with lipid RNA nanoparticles in terms of percentage of cells transfected. However, when you then go in vivo, and this shows you some of the difficulties with doing some of these experiments, with the difficulties in doing cell-based experiments, then going into a mouse before going into a human. And what we saw, in fact, was that the most potent polymers in mice were actually, it was a commercial polymer, which we effectively wanted as our control. So that was very disappointing because we'd hoped that our investigational polymers, which work so well in cell lines, would be better than in a mouse. But actually that wasn't the case. We found the commercial polymer actually performed better. There was still some level of transfection, but we're now looking at why that might be. And of course, these particular mice may not be a good model for a human target. So you often have to temper your studies with trying to work out what's the most realistic model. So a difficult take-home message for this one, but whenever you come on to immunological studies, the answer is usually it's complicated. And one of the things we're trying to dissect is structure function studies to look at what polymers do what and why. We've been working very hard on this, and we've filed some IP, and I can't talk about that at the moment. But the interesting thing here is that we are beginning to see some correlations between in vitro studies and in vivo. So let's go on to the next part of the talk, the second half of the talk, and that's looking at formulation science for cancer drugs. And as I said earlier, many drugs for cancer have a very narrow threshold between efficacy and tox. And one of the things, of course, is that if you use a polymer carrier, you reduce systemic exposure of cytotoxic drugs, and so that can be beneficial for the patient. You don't go above the therapeutic window. But the other thing that's interesting is that some nanoparticle carriers can enter cells via different mechanisms to small molecule drugs. So they don't go in through conventional cation or anionic transporters. And that can actually give a change in drug efficacy dependent on the route of internalization. So if the drug goes in through a different door, if you like, into the cell, then it can alter some different pathways. And this is methotrexate. And you can see in this case that the polymer delivered methotrexate is actually much more potent than the free drug itself, which is usually the other way around. You don't normally expect it to be quite as effective if the polymer doesn't release all the drug. So there are some interesting points on this. Now, we've been looking at these for triple negative breast cancers. And the reason we've been doing that is because currently triple negative breast cancers have a very, very poor prognosis. And the standard of care methods really are not great, often acute and long-term side effects. There's usually a high relapse in TNBC patients, and there's a range of different reasons why that might be the case. However, one of the things is that certain triple negative breast cancer cells do have this increased intracellular reducing agent glutathione and all system enhanced proteolytic enzymes, and that's one of the things that they exploit to get their competitive advantage. But then we can start exploiting that by looking at designing our polymer carriers to take advantage of some of the changes in biology in the cancer cells as opposed to normal cells. But for all of this, you require polymers which are stable after systemic injection and which can release drugs following accumulation in the tumor. So you want to have something that can travel around the body safely, but then only releases its drugs on the right side. And this is where we started this investigation. We started this about actually 5 years ago now. And we assembled a team to do this. I'm only going to talk about a small part of this work today, but essentially we made a whole bunch of polymers with different sizes and different architectures, but also with the drug compound attached via a shift-based linkage and with some reducible branching agents, and we also put in some reducible components in attaching to the drug. So we had reducible, non-reducible, and so on. And the idea was that we'd make polymers of a certain architecture, and then over time they're designed to degrade into smaller fragments and release the drugs. So the idea is they don't accumulate in the body, and they release not only the drugs, but they also break down themselves. So we want to look at architecture and chemistry simultaneously. And so just to show you that we made what we wanted to make, we made a bunch of polymers, stars, hyperbranch, linear polymers, micellar type systems, degradable and non-degradable. And we had a range of sizes. So you can see in the DLS that we had a range of different sizes. And of course, we confirmed it by TEM. One of the things that's really important is to do orthogonal methods, so you don't rely on just one characterization technique. And we essentially saw that if we wanted to work for our reducible polymers, they disassemble and also they degrade in the presence of this intracellular reducing agent, glutathione. And this occurs very readily with some polymers, less so with some of the others. And interestingly enough, the micellar type polymer, perhaps because the disulfide bonds were deep in the interior, essentially were quite stable, whereas the other ones, branched and so on, were less stable to glutathione. So there's some interesting things in terms of architecture and how these things degrade. But there are also some interesting things in terms of how the polymers are distributed in the body. And so we dosed in various mice with labeled compounds. And I'm just going to walk you through this graph here. And I'm going to guide you through. So on this side, the left-hand side of graph, as I look at it, we have polymers going from 6 to about 8 nanometers. And then on the right, we have polymers which were larger in size. Now, this linear polymer, in fact, probably assembled into aggregates as well, hence the 30 nanometer size. What was interesting were these two in the middle. So you can see one which has around 10 nanometers and one around 12 nanometers. But one went to the kidneys and one went to the liver, predominant. So they looked pretty similar. One was a hyperbranch, one was a star, and yet their distribution was very different. We found they were both effective in destroying 3D cancer tumor, 3D spheroids, so models of tumors, but they're also very effective in an orthotopic model in vivo. So they were better than the free drug. If I look at the free drug here in orange, then our two polymers in green and blue. So this is as close to a model as we could get in a mouse model anyway of triple negative breast cancer. This was not a xenograft, this was an orthotopic model, and we saw essentially very little increase in tumor volume, even when the free drug was unable to prevent tumor growth. We saw no weight loss, and we saw greater tumor reduction in terms of mass, and here's the pictures to show it. But this is kind of odd because we didn't really know why that should be, and we've been trying to unpick that in recent months. We had a big in vivo study ready to go just before COVID. So essentially, we had to make the polymers all over again, do all the studies all over again. So it's taken us about 2.5 years to go from that previous slide to the next two or three slides, which are the conclusion to this talk. So here are the representative polymers again. So this is hyperbranch, about 52 kilodaltons. Size from DLS is about 10 nanometers. Now, we do get some self-association. There may be some kind of discotic behavior when we see these in the TM. Likewise with the star, this is a higher molar mass, but the diameter, light scattering, pretty similar, 10, 16 nanometers. So remember, these were represented the ones that went to the kidney. These were representative of the ones that went to the liver and spleen. Now, as I say, they weren't exactly the same sizes. We're never able to make them exactly the same size as we had done before. But if you look at their distribution over time in the body, so this is their residence time in the blood, then you can see that one is essentially gone after 24 hours. The other one actually is still sticking around, okay, after 24 hours. And we still see a signal after 48 hours. And you can see that very clearly. There's not so much of a signal here, but by 48 hours, this star polymer is still going around the body. So quite a marked difference in the distribution. And this is work, really fantastic work by Cara Maloney, Fatima Maradnia, and Rob Kavanagh. So been doing this recently. But what's really interesting now is we did some histology, and we looked at literally hundreds of tissue slices from tumors and all the different organs. So it's a real tour de force from these people. And what you see, quite interesting here. So this is from the tumors, that these polymers delivered more doxorubicin. So there's more of the green signal here when the polymers carried the drug than there was of the free drug in the tumors. So even if you don't see a tumor shrinkage, there was definitively more drug actually in the tumor tissue. And you can see that in the graphs here. And then the story gets also going very, very interesting in the sense that we saw differences in terms of the distribution. As I mentioned before, we saw some of these polymers accumulating more in the kidney versus the liver over a particular time. Now, when we redid these studies, we see again, this case, there's a lot in the kidney, and this case, there's a lot more in the liver. So that's kind of interesting. But what was also intriguing was when you start looking at apoptosis in tumors as opposed to the other organs, and again, we really don't know why this is the case. So cell death, so apoptotic markers in tumors were much, much higher for the polymers than they were for the free drug. But also they were much lower in the heart and the liver. So the polymers are much less toxic in the heart and the liver than the free drug. Now, cardiotoxicity is a limiting factor for doxorubicin. It's a limiting factor in the clinic. So this would be a tremendous advantage. We didn't see a huge difference in the spleen, and we're still trying to work that out. But nevertheless, in terms of therapeutic window, this should be an enormous advantage. Okay, so just to conclude, as I would say this to an audience of polymer scientists, it's central to materials development. So synthesis is central to the materials development for biomedical applications. And looking at how polymers are structured, how they interact with RNA or interact with cancer drugs is really important in terms of whether our formulations can be stored for a long time, whether they can be injected into particular sites of our particular routes. But we still don't understand a lot of the components of how polymers and RNA assemble, how they disassemble, and certainly how they behave in vivo. That's still a major challenge. I mean, it's taken us nearly 9 months to look through all the tissue samples that we've been obtaining from one in vivo study. So we're now trying to look at how we make these polymers in a rather more sustainable method. And like others, we're doing a lot more in flow and a lot more of fluidics now. We're doing a lot with engineers, but also we're looking at social scientists about how tolerable our systems might be. I'd also say the social component is really important. We have a big bunch of people from all over the world, and I say that collaboration is essential for progress. Just want to give a few thanks. There's a bunch of people. This is the group at the end of last year. This group just before COVID, but we work with people all over the world, and we're funded by some very generous people. And at that stage, I'd just like to thank very much the organisers, and I'm very happy to take any questions.
Okay, thank you very much, Cameron.